Welcome to this lecture on parasitology. Today, we are going to explore a fascinating and formidable microscopic invader known as Hymenolepis nana, or the dwarf tapeworm. Imagine a tiny, unwanted guest living inside your digestive tract that is so small you would never notice it without a microscope. Understanding this tiny invader is the first step to mastering human parasitology and preventing widespread infections. We will cover its biology, how it spreads, and even use mathematical modeling to understand its impact and treatment. Let us dive in. To begin our journey, let us meet the parasite itself. Hymenolepis nana is commonly referred to as the dwarf tapeworm because it is the absolute smallest tapeworm that infects humans. While other tapeworms can grow to be several meters long, this particular species typically measures only 15 to 40 millimeters in length. Despite its incredibly small size, it belongs to the class Cestoda, which includes all parasitic flatworms. Do not let its microscopic stature fool you. What it lacks in size, it more than makes up for in its ability to rapidly multiply and cause significant health issues within the human gut. Now that we know how small it is, let us look at how it is built. Structurally, the dwarf tapeworm is highly specialized for survival inside the harsh environment of the human digestive tract. It consists of a tiny head called a scolex. This scolex is armed with a retractable ring of hooks, which act like microscopic grappling hooks to firmly grip the intestinal wall so it is not washed away. Following this head is a long chain of segments called proglottids. These are essentially reproductive factories, packed with eggs, which break off and release their contents to continue the parasite's lineage. Understanding its anatomy leads us to its most terrifying superpower, its life cycle. What makes Hymenolepis nana biologically exceptional is that it is the only tapeworm capable of completing its entire development, from egg to adult, within a single host. Most parasites require an intermediate host, like a pig or a cow, to develop before infecting humans. This tapeworm completely bypasses that need. Furthermore, it can undergo internal autoinfection, meaning the eggs hatch inside the same intestine without ever leaving the body. This direct cycle allows the parasite population to explode rapidly, turning a single swallowed egg into a massive colony. This explosive reproductive capability brings us to why this parasite is a massive global health concern. Hymenolepis nana is actually the most common tapeworm infection in humans worldwide. It primarily affects children in areas with poor sanitation, spreading rapidly through fecal-oral contact or contaminated food and water. Because of its auto-infection cycle, an untreated infection can last for years. This persistent burden causes chronic malnutrition, severe abdominal pain, and debilitating diarrhea in vulnerable populations. By grasping these biological dynamics, medical professionals can intervene early, stop outbreaks in crowded environments like daycares, and ultimately protect community health. To truly understand the severity of this public health threat, we must move beyond biology and look at the numbers. We need to quantify exactly how rapidly these parasites can contaminate the environment. By calculating the total daily egg output of an infected individual, epidemiologists can estimate the environmental contamination risk in a specific community. This mathematical approach allows us to predict the scale of an outbreak and allocate medical resources effectively. We do this by looking at the number of adult worms, their shedding rate, and the egg capacity of each segment. Let us look at the formula used for this. Building on that concept, here is the mathematical model used to calculate environmental contamination. The formula is E equals W times S times C. Let us break down what each symbol means. E represents the total number of eggs shed in the feces per day. W stands for the total number of adult worms currently living in the host's intestine. S represents the shedding rate, specifically the number of egg-filled segments, or gravid proglottids, dropped by a single worm each day. Finally, C denotes the average capacity, which is the number of eggs inside one single segment. If the worm burden, W, increases due to autoinfection, the total egg output, E, will scale up linearly, drastically increasing the chance of infecting others. Now that we have our formula, let us apply it to a practical scenario involving a heavily infected patient. Suppose a child is infected with 50 adult worms. Each worm sheds 4 segments per day, and each segment contains an average of 200 eggs. We want to find the total daily egg output. First, we substitute our given values into the equation, replacing W with 50, S with 4, and C with 200. Next, we multiply the 50 worms by the 4 segments, giving us 200 total segments shed every day. Finally, we multiply those 200 segments by the 200 eggs inside each one. The result is 40,000 infectious eggs shed into the environment every single day. This massive number clearly illustrates why thorough hand washing is absolutely critical to stop the spread. Seeing that massive number of eggs brings us to an urgent question. How do we stop this? Once diagnosed, a patient must be treated promptly to eliminate the parasite and break the dangerous autoinfection cycle. The standard medical treatment for Hymenolepis nana is a highly effective drug called praziquantel. This medication works by paralyzing the worms. Remember those microscopic hooks we discussed earlier? When the worm is paralyzed, it loses its muscular grip on the intestinal wall. Once detached, the paralyzed worms are simply expelled from the body through normal digestion, effectively curing the infection. To administer this treatment safely, we must calculate the exact dosage. Here is the standard pharmacological weight-based formula. D equals WP times RD. Let us define these variables. D represents the total required dose of medication in milligrams. WP is the patient's body weight in kilograms. RD is the prescribed dosage rate, which is the amount of drug required per kilogram of body weight. This formula ensures the drug concentration is high enough to paralyze the tapeworms without causing unnecessary toxicity to the child. If the patient's weight increases, the required dose increases proportionally to achieve the exact same paralyzing effect on the parasites. Let us put this dosage formula into practice with a real patient. The recommended single dosage rate for praziquantel is 25 milligrams per kilogram. If we have a pediatric patient who weighs 24 kilograms, what is the total required dose? We start by substituting our known values into the equation. We replace the patient's weight with 24 and the dosage rate with 25. Now, we simply multiply these two numbers together. Multiplying 24 by 25 gives us a total of 600. Therefore, the patient requires a single dose of 600 milligrams of praziquantel. This precise calculation is vital because underdosing would allow surviving worms to continue their autoinfection cycle, while the correct dose breaks their grip entirely. Now that we have successfully treated the patient, Let us look closely at the microscopic culprit we are fighting. This diagram shows the morphology of a single Hymenolepis nana egg. Notice the protective outer shell and the inner membrane, which shield the parasite from the environment. Inside the inner membrane is the oncosphere, which is the actual embryo. You can clearly see the tiny hooklets inside the oncosphere. These will eventually develop into the hooks on the adult tapeworm's head. Finally, observe the polar filaments extending between the membranes. Recognizing these specific structural features under a microscope is exactly how medical professionals diagnose this infection in the lab. We have covered a lot of ground today, so let us summarize our key takeaways. First, we learned that Hymenolepis nana, the dwarf tapeworm, is unique because it can complete its entire life cycle within a single human host, using autoinfection to rapidly multiply. Second, we saw how mathematical modeling of daily egg output reveals the staggering scale of environmental contamination, emphasizing the need for strict hygiene. Finally, we learned how to calculate precise weight-based dosages of praziquantel to safely paralyze and eliminate the parasite. You now have a solid biological and mathematical grasp of the world's most common tapeworm. Keep this knowledge handy as it forms a crucial foundation in medical parasitology.